everyone. This is Kiefer. I am the TA for one of the TAs for Physiology, and Grace is my partner this semester. Some of you may know me, some of you may not. Unfortunately, as you guys saw Candy's email, weather permitting, didn't really allow school to happen today. So to make up for this, Grace and I are going to kind of do a voiceover presentation of the highlighted aspects of this lab that we want you to know. It's not going to go into everything, but it's going to go into the major points that we kind of would have covered in our intro anyway. So make sure that you have your lab book out, you're kind of following along, seeing what we're talking about. And then, like Candy mentioned, it would be really great if you guys could go in and look at those slides, because they are quiz material. Make sure you're going through those activities, yada, yada, yada. Okay, so I'm going to start off with the first portion of the lab, which is your introduction to microscopes. I'm sure most of you have used a microscope, but we do have to do that annoying little go over to make sure everybody knows how to care for them. In one of the first pages of your lab manual, there's a diagram of a microscope. Review, refresh, learn all of those tools on the microscope, because they could be quiz material. Make sure you know what they do. If you don't know or aren't sure, ask somebody in the lab. It's kind of hard to do without a video or without you working on the microscope itself. So just kind of review it, refresh it, look at it. So one important thing that we want you guys to know is that you have your objective lenses, which are your 4x, 10x, 100x, but that's not your magnification. You have to calculate your magnification. In this case, your eyepiece has a 10x, so in order to calculate the magnification that you're looking at, you have the strength of your eyepiece by the objective lens. So, for example, if you're looking at things under the 4x, you take your 4x and you multiply it by your 10x eyepiece to get 40 times the magnification. And that's something you guys will have to know. Make sure you're fiddling around with your adjustment knobs to get things into focus. And an important terminology you guys will have to know is parfocal. It just means that you can look at something under, say, 40 times the magnification, switch to the 10x objective, and things will be still in focus, which is cool. Okie dokie. So, another thing that you guys are going to have to fiddle around with is your depth of focus. It's just basically, it's a 3D image kind of that you're looking at with those thicker samples. So you can focus light onto the sample, or you can focus light through the sample. Each will kind of give you a different view of the different components of the slide. So make sure that you're fiddling around and working with to see the whole slide and not just some of it. Okay, I think that's all for the microscope. Moving into the histology portion. Histology is the study of tissues and the cells and the components that make up those tissues. So it kind of comes in like two parts. You have like a cellular type of tissue and an acellular type of tissue. Just as it sounds, cellular tissues have more densely packed cells in them than acellular tissues do. Acellular tissues have way more of that ground substance. For example, like proteins, globulins, electrolytes, all that kind of stuff. It's more of that liquid ground substance than it is that dense packed cell type. There's 4 types of tissues that we're going to go over. I'm just going to briefly cover epithelial, and Grace will take over connective, muscle, and nervous. So for epithelial tissue, it's cellular. It's densely packed with little ground substance or extracellular fluid. You find it, surprise, on your skin, your glands, internal and external membranes. It's just kind of that protective lining. To break down epithelial tissue even further, you can have different shapes and different types of layers. So if it's just one layer and it's just called simple enough, And if you have more than one layer of cells, it is called stratified. Additionally, there's different types of shapes of cells. So you have cuboidal, which just are as they sound. They are cuboid shaped. You have taller columnar, which are like rectangles. And then you have squamous cells, which are like pinched at each end. And then building on that, you can have a simple columnar, a stratified columnar, simple cuboidal, that kind of stuff. This lab doesn't go into generally like where in all parts of the body you're going to find, simple cuboidal or stratified squamous, that kind of stuff. It is just going to give you some pointers. So make sure you look at that. Stratified squamous is your most protective covering. Its lines are skin. It's durable. And then simple squamous lines your cardiovascular system on the inside towards the lumen, and it's actually the most fragile. A lot of those cuboidal and columnar cell types are found in glands that need to produce lots of things and secrete them. So, yeah. If you guys have any questions about anything that I've covered, make sure to shoot me an email. I'm not in Idaho right now, but I will have access to my email, and I will hopefully be back by our lab next Wednesday for those who are in my lab at 2:30. But I will have my email, so shoot me a text or shoot me an email anytime you need. Alrighty, thanks guys.
Okay, so we're going to continue on with connective tissue. Something to note is that it's acellular, meaning that instead of having an aggregation of many cells, you're going to have a higher amount of ECF in comparison to cells. There are two ways to classify connective tissue. You can do it by protein fibers, or you can do it by the viscosity of ECF. So if we look at protein fibers, there are three different types that I've written here. There's collagen. This is straight, thick protein fibers. They provide strength. The following one is elastic fibers. So these are wavy, branched, do not provide that much strength. However, they do stretch. And the last one is reticular fiber. These are straight and branched. They're semi-strong and provide a little bit of stretch. So the reticular one has aspects of both collagen and elastic properties. So another way you can classify it is by the viscosity of the ECF or ground substance. So let's say you had a very watery ground substance. This would signify that we have a fluid connective tissue. So example of this would be blood or lymph. And typically fluid connective tissue are used to transport nutrients, oxygen, and other gases throughout our body. The next one I have here is fibrous connective tissue. And typically the ground substance here is a very syrupy type of ground substance. So this can be found in ligaments and tendons, and they typically provide support as well. And the last one I have here is where we're going to concentrate most of our time on. So the last one is the supportive connective tissue. This is very thick and solid. So a place where you might find this, like I've written here, is cartilage and bone. So we'll go ahead and talk a little bit about cartilage and the different types of cartilages we have in our bodies. So the first one I have here is hyaline cartilage. And in hyaline cartilage, you have a lot of collagen fibers, you have a lot of chondrocytes. It provides protection for our body. So an example of where you might find this is in the thoracic cage and in articulations. And then hyaline cartilage, a really unique thing about it is that it can later become ossified, meaning that it can later become bone. So it's a unique aspect of the hyaline cartilage. And then the next one I have here is elastic cartilage. So when you think elastic, you probably have elastic fibers. So this does have elastic fibers and has a very jelly-like ECF, or extracellular fluid. And it has some chondrocytes. It's very flexible, a place where you might find elastic cartilage, for example, in your the lobe of your ear and your outer nose, places like that. And then the last one I have here is fibrocartilage. So this consists of both collagen and elastic fibers with some reticular fibers. It has few chondrocytes. So these can be found in intervertebral discs and in the pubic symphysis of the pelvis. So the next tissue I'm going to be talking about is bone. So this consists of a solid extracellular fluid or ground substance, and it has a lot of collagen fibers, meaning that it's very strong and supportive. And so we'll go ahead and talk about two specific types of bone tissue. So we have compact bone tissue. This is very organized with collagen fibers in concentric circles, and these are in the lamellae. So this is essentially just ossified hyaline cartilage like we talked about before. Because it's very organized, you would consider these to be osteons. So there's typically one osteocyte per lacunae, and this gives it a lot of strength and a lot of protection to keep it together. The next one we have here is spongy bone tissue. This is used in areas of less bone stress to decrease weight of the skeleton, so there aren't that many osteocytes here. It's very solidified. There's a lot of calcified ECF and collagen fibers, but they're randomly put in the ECF. It's not as organized as the compact bone tissue. These don't have osteons because it's not organized like the compact bone tissue. So this is called, instead it'll be called trabeculate, meaning unorganized. So here I have some pictures. This is of cartilage, and it shows you the lacunae and how it has the chondrocytes. And essentially what a lacunae is just a small cavity that contains either, in this case it would contain chondrocytes. However, when you're looking at, this is compact bone, you'll see that it's very organized and has osteocytes instead. So I'll go ahead and move along to muscle tissue. So this is cellular, just opposite of the connective tissue. So this has lots of aggregations of cells. There are three different types of muscle tissue that we'll be talking about. We have skeletal muscle, cardiac muscle, and smooth muscle. So typically muscle tissue is specialized to contract and shorten within the influence of electrical signals. So skeletal muscle, this is voluntary, typically attached to your bones and skin, and helps move skeleton at articulations, and like I said, can be voluntary movement. It's very striated, meaning that it's very organized. So this has the same or similar ECF of the compact bone, and the cells within themselves are very long and thin, so they represent a fibrous cell. And then right here we have cardiac muscles. This is involuntary. It's less fibrous than skeletal muscle cells. The cardiocytes are not branched and not linear, and they join together to form a network pattern. And these are only found in the heart, where it's involuntary, controlled by the CNS. And again, this is striated, meaning that it's very organized. And the last one I have here is smooth muscles. This is non-striated, meaning that it's not very organized like skeletal and cardiac. It's involuntary. You'll find this in your digestive system, so it'll help move food along your digestive tract. Typically, these are found where it can help move materials, so like I said, the digestive tract, or it could be blood, air. So the cell shape is actually fusiform in shape, unlike the skeletal muscle, which is fibrous, which represents a fibrous cell. Alright, well, I think this is it, so we'll go ahead and wrap it up here. Like Keeper said, make sure you do all the activities. All the slides are definitely quizzable. Everything in the lab manual is definitely quizzable. So go ahead and make sure you understand all the different aspects about the different tissue types and how to classify them. And yeah, make sure you really are thorough on those activities, because those will help you complete the quiz next week. If you have any questions, feel free to contact any of the TAs or Candy or Tara. That way you can do well on your quiz next week. Alright, well, good luck. Study on.